Hi, this is Maggie Vires, and this is my exam 3 talking about how DNA is replicated in microbes. So, DNA can be replicated two different ways it just depends if it's a eukaryotic microbe or a prokaryotic microbe, and they have a lot of similarities, but they are different in their shapes. A eukaryotic microbe is a double helix DNA structure, it's linear, whereas the prokaryotic is circular, it's the circle of chromosomes, whereas the double helix is like a ladder. And the double helix is made up of nucleotides, and these have three different parts. They have a phosphate group, a deoxyribose sugar, and a nitrogenous base. So the backbone of this is called the sugar phosphate backbone because of these components. And these bases, like adenine and thymine and guanine and cytosine, these are the same in both prokaryotic microbes and eukaryotic microbes. So the base pairing rules are the same, and they're held together by peptide bonds. And the other thing that is the same is the anti-parallel strand. So it goes from the 5 to 3 direction, and then the other strand goes in the 3 to 5 direction. And this is essential for DNA replication and to help make proteins. So what we might see in prokaryotic microbes that is different from eukaryotic is plasmid DNA, and these are just small circular chromosomal DNA molecules. So these are not found in eukaryotic molecules. And that's pretty much it for that. So here's how DNA is replicated. So when it needs to be replicated, there is an important first step is unwinding the strands of the double helix. So this will occur at replication origins, and replication origins different between eukaryotes and prokaryotes. The prokaryotic origin of replication, there's only one, and this means there's only 2 replication forks, whereas in the eukaryotic one, there are several. There could be up to, I don't know, even like 100 or even just a few, It's not limited to a certain number. There are many origins of replication. And this means, so if there's like 100 origins of replication, there's 200 replication forks. But they will occur, the unwinding occurs at the replication origins, and the helix will uncoil because of helicase, which is an enzyme. And the hydrogen bonds will begin to separate, and it's like the helix is unzipped. And DNA polymerase 3. This is an enzyme, and it is part of this process, so it needs a primer to begin the process, and it only builds on into the 5 to 3 direction. So we're going to go into this a little bit next and how it plays a role. And the replication, it begins at the replication fork, and it's kind of Y-shaped, as you see in red. And this is where the DNA is opened and copied. So in circular bacteria, there are 2 replication forks, and in eukaryotic, there are many. So each strand of DNA is used as a template. So the old strand is used as a template, and the bases will attach and use that template to make a new identical template, creating 2 new strands from the one strand before. So they consist of an old strand and a new strand. And this applies for both prokaryotes and eukaryotic microbes. So here's some key enzymes that I just talked about, the helicase and DNA polymerase, but I'm going to go into them a little bit more because they are very important. So each enzyme has its own specific role, and it signals for different things. So helicase is signals for the DNA helix to unzip, and then primase is used to lay down a short RNA primer, and this helps get the process started. So that's towards the beginning, it primes it. And then DNA polymerase 3, this is the main enzyme that synthesizes DNA, and like during replication, and it works on both the leading and the lagging strand. And well, it works on the leading strand continuously and the lagging strand discontinuously, so that's important. It makes very few errors. Whereas DNA polymerase, it's not as important. It only removes the primer and fills in gaps within the DNA. So its job is really just to help proofread and clean up. And ligase, this is used to seal in the cracks between the Okazaki fragments. And then we have the topoisomerase 1 and 2. These are used to prevent DNA from over-twisting, so it's towards the end of the process, and it just helps it get back on track. So now we're going to talk about proofreading and accuracy. So when the DNA replication messes up, it's able to fix itself through different enzymes like the DNA polymerase 3 and 1. So an occasional mispairing is called a mutation. So it sometimes happens, but it's a very small chance. Like the rate in bacteria, as you can see, is like 1 in 1 billion. And we also read that E. coli produces 750 in a second. So if it's producing almost up to 1,000 in a second and it's only making an error every 1 in 1 billion, it's pretty accurate. So we have two proofreading enzymes. We have DNA polymerase 3, and we've seen that this one already plays a stronger role, and it can take out the wrong base and substitute it with the right base. And then DNA polymerase 1 can also proofread the bases and then repair them as well if the DNA is damaged or if they're incorrectly placed. So I'm going to talk about semi-conservative replication. This is what I talked about a little bit earlier, that each DNA has an old strand and a new strand, and the strand before it is used as the template, which makes sure it gets the original code, and it will continue to have that code throughout generations. So this is called semi-conservative. So like the blue strand is the old strand, and then the red strand is the recently generated strand with the same code. So there are some limits on replication. So DNA polymerase 3, it can't start just on its own. So it can only begin and add to an existing chain, and it only builds in the 5' to 3' direction. And then replication forks, these are when the strands are unwound and copies. So the circular DNA has 2 forks, like I said before, and then the eukaryotic has, say, 100. So then it would 100 replication origins. Then it has 200 forks. Okay, now I'm going to talk about the leading and lagging strand, and this picture really dives into the replication process. So because DNA runs in opposite directions, the replication isn't the exact same on both sides. So the leading strand and the lagging strand, they run in opposite directions and they won't be identical. So there are similarities between the leading and lagging strands in eukaryotes and prokaryotes. So they're both synthesized in the 5' to 3' direction, and the lagging strand is synthesized discontinuously into short Okazaki fragments, and this is opposite of the helicase movement. And they both require an RNA primer from the enzyme primase. And some differences between the eukaryotic and prokaryotic leading and lagging strands. So in prokaryotic, they use DNA polymerase 3 for the leading strand and the lagging strand together, and DNA polymerase 1, this is used to remove the RNA primers and replace them with DNA primers. But in eukaryotic microbes, it uses different enzymes. It was a different type of polymerase, whereas in prokaryotic, it's polymerase 3 and 1. So for replication of linear DNA, so this is talking about eukaryotes. This is going into the topoisomerase, and its main function, it is to help relieve the stress from the twisting, and it just kind of helps get the DNA back on track after unwinding and being extended. And I'm also going to talk about the eukaryotes will replicate the DNA in a way that's similar to bacteria, but their DNA is linear and not circular. So they require many more polymerases instead of just a couple. And this makes sense because bacteria only has one replication of origin, where eukaryotic has many. So now I'm going to talk about telomeres. So these are at the ends of the chromosomes, and they're like protective caps. So over time, when the cell divides, these will become shorter and shorter with age, and over time they become too short and will send out a signal and trigger apoptosis, which is a programmed cell death. But there's also the issue, this is a result of the cell copying, because DNA polymerase cannot copy the 3' end. So over time it gets cut off, and there is a little bit of portion that doesn't make it into the copied strand. So